Hello. Naturally occurring extra-chromosomal DNA molecule can be modified to obtain vectors. In the previous session, we have discussed about plasmid vectors, and we see that the naturally occurring plasmid molecules were combined and modified to get PUC18 and PBR22. In this session, I am going to talk about viral vectors. The viral vectors are obtained by modifying the virus genome. And when we are comparing the features of the plasmid vectors with the viral vectors used to have better better properties. So we begin our discussion with the lambda vector. Lambda vector or lambda phage is lambda vector is actually originated from lambda phage. Lambda phage is a coliphage. That means it is affecting coliform bacteria. Here is the image of the lambda phage. The lambda phage is having a hexagonal head and the DNA is actually packaged inside that head region. Then it has a tail. So during the time of infection, this tail is actually aiding the absorption of these viral particles into the coliform bacteria. So after absorption, it will be injecting the viral genome into the bacterial cell. Inside the bacterial cell, there are two possibilities. Either it can undergo a lytic phase cycle of growth or it can go for a lysogenic phase. The lytic phase involves rapid multiplication of the uh, phage DNA. So it will begins with like the production of the viral proteins. The production of viral proteins leads to the replication of the viral genome, and this replicated viral genome will be packaged into the structural genes of the phage, and finally it will disrupt the cell and new viral particles will emerge. So that's the lytic cycle. But in case of lysogenic cycle, this type of infection do not happen. What is happening there is that the DNA is getting integrated into the bacterial genome and it will be silent or dormant for a very long time. So the lambda phage can either undergo a lytic phase cycle of growth or lysogenic cycle of growth depending on the nutritional characteristics and the properties of the bacterial cell. So one more thing, the growing the viruses is a bit difficult than growing the bacteria. So for the growth of a phage or the bacteria virus, we need living cells. So in the case of lambda phage, we need Escherichia coli. So what we do for this culturing the lambda phage is the top agar culture. So the top agar culture is doing like in this way. First we will have the agar plate, and on top of that we will add the top agar. Top agar used to have lower agar concentration, so it won't be that much hard. So we'll be mixing the top agar along with bacteria and the lambda phage, and after mixing it will be poured into the normal agar plate, and after the pouring, bacteria starts to grow and the virus starts to infect the bacteria. So the viral colonies or viruses infected cells will form a clear region in the bacterial lawn. These regions are called plaques. So the bacteria, when we are growing a bacteria, we'll be seeing the colonies, but when we're growing a virus, we'll be seeing the plaques. So each plaque representing the virus originated from a single mother virus or a single virus. So coming to the genome of the bacteriophage lambda, the lambda genome is a bit bigger. Its size is about 48,502. We can say it's 48.5 kilobase pairs. And one other feature is that inside the lambda head, it is a linear genome. The linear genome is having two different 5' end and a 3' end. The 5' end, 3' end is having an overhanging, and this overhanging of the 5' end and the 3' end are complementary in nature. This overhanging is called a cos site or cohesive site. So inside the head, it acts as it is a linear DNA, but after the infecting a bacteria, the DNA will be transferred into the cell. But inside the cell, due to the property of these cohesivents, they will come together and bind to form a circular DNA. So inside the lambda phage, it's a linear DNA, but inside the bacterial cell, once it reaches the bacterial cell, it will become a circular DNA with the help of host site. Okay. One other feature of the lambda genome is like the functionally related genes are clustered together. The genes which is responsible for the DNA replication is in one place. They all genes are situated near to each other. And the genes for the lysogenic cycle is actually packed together, and genes, the structural genes, which is required for the reproduction or the making new virus particles, is also packed together. So it's like different regions for different functions. So that's a very good feature when we are trying to make a vector out of it. So this is a structure of the lambda phage. So lambda phage size is 48.5 kilobase pairs, and here you can see these genes are for the DNA replication, then this is for the recombination or lysogenic cycle, and this one is the structural genes. And here we have this cos site consisting of two overhangings, 5' and 3' overhangings. These overhangings are actually complementary in nature. That's how it will become circular inside the bacterial cells. Here is the linear representation of the same. In the both end we have the cos site, and genes are clustered together. So as I said before, the virus after the infection, it can either for a lytic cycle or lysogenic cycle. If it is a lysogenic cycle, DNA will be integrated into the bacterial genome, and as the bacterial genome replicate, the DNA also get replicated. But in the lytic cycle, what's happening is like there is a drastic change in the bacterial system. The bacterial system will be controlled by the virus, and the virus will get replicated and finally rupture the cells to release about 100 viral particles from cell. And one of the main factor which is actually controlling the adoption of lytic or lysogenic cycle is the C2 gene. So after the infection, if the virus is taking a lytic cycle, there is different there's a series of transcription. Okay, there is a series of activities it is doing in order. So first process is called early transcription. In the early transcription, the early genes get transcribed, which includes N and CRO. Along with that, a repressor is produced, CI repressor or C1 repressor, sometimes people say. This repressor is actually preventing the lytic cycle. Okay, so it's actually aiding the lysogenic cycle. That's the early transcription. Then if it's past the early transcription, it goes to the delayed early transcription. It is actually governed by protein N, which is produced in the early transcription. It's actually increasing the expression of proteins which is responsible for the DNA replication, the replication of the lambda genome. Then it goes for the replication stage. During the replication stage, DNA will be getting replicated. Initial replication is from the origin of replication, but later it adopts a rolling circle model of replication. So rolling circle model of replication will form a concatenameric DNA, one single DNA molecule which is containing multiple lambda genome. And finally, we have the late transcription. Late transcription is actually initiated by CRO gene, and it produces the head and tail structural proteins. And after the late transcription, the replication product, that is the concatenameric DNA, and the structural proteins are combined to produce a viable phage, and the which ultimately leads to the lysis of cell and release of the viral particles into the medium. So this is the representation of the rolling circle replication. So this is a double-stranded DNA. Then it will form a nick over here, and from this nick, this DNA molecule is displaced. Okay, actually it is circulating. So this blue strand is displaced, and it is replication is taking place in this direction, and it will never end. So as the replication proceeds, this length of this product will be increasing. So you can clearly see here our template DNA is a circular DNA. Okay, but our product is a linear DNA. One other feature is that this linear DNA will have multiple viral genome. This blue starting from here to here is actually one viral genome. And secondly, we have getting other viral genome. And this process, the rolling circle replication, is continuing forever. Not forever, but continuing for a very long time, so that what we get is a long DNA with a multiple viral genome. So once we have the DNA and the late transcription is producing head and tail proteins. There is a process of phage assembly. Phage assembly is done in different steps. So we have the concatenameric DNA. The concatenameric DNA is having uh, multiple viral genomes in a single linear DNA, and we can see the cos site here, and here is an empty head. So head with nothing. So this empty head combined with the concatenameric DNA, and head has some endonuclease activity, so it will cleave the DNA at the cos site. Okay, so out of this concatenameric DNA, one piece is cut and it is inserted into the head. Once head attached with the DNA, it will become a stable head and tail is getting attached. So only to the stable head, that is the head which is attached to the DNA, will get the tail. So then we have a infectious phage or complete virion. So this can be released into the medium and it can infect another bacteria and start another circle of replication. So this wild type lambda phage is having so many features which can be used as a vector, but use is limited because it do not have unique replication, unique recession nucleus replication site. And one other factor is like the head of the phage, lambda phage, cannot like it has a limit in the DNA, how much DNA it can carry. So it can carry about 78 to 105% of the real genome. The real genome size is actually 48.5 kilobase pairs. But it can carry, if the size is up to 51 kilobase pair, the head is okay. But if your DNA is longer than 51 kilobase pairs, it cannot accommodate. There is not enough space for that. And in the lower side, the size can be up to 37 kilobase pairs. If a DNA is 37 kilobase pair, it can be packed into the lambda head. But if the DNA is not 37 degrees Celsius, maybe 35 degrees, 35 kilobase pairs length, it cannot be fit into the lambda head. So in order to fit a DNA into the lambda head, the size should be between 37 to 51 kilobase pairs. Okay, the actual size is 45.5, but there is some flexibility. So any DNA with a size between 37 to 51 kilobase pair will fit into the lambda head. So what we can do, we can actually modify the lambda phage DNA by eliminating the restriction nucleus recognition site, and we can actually remove some of the regions, non-essential regions. As I said before, maximum capacity is the 51 kilobase pairs. Actual size of lambda genome is 48.5. So what we can add is about 2.5 kilobase pairs. That's nothing. That's a small amount of DNA. If you want to add more of the DNA, we can remove non-essential parts from this genome. One such example is the regions for lysogenic cycle. So for a vector, we don't need the lysogenic pathway to be there. So what we can do, we can remove the genes which is essential for lysogenic cycle from the lambda genome, and we will get a smaller virus genome, so that we can add more of the genome. To the original viral genome, maximum its size is 48.5. What we can add is up to 51 kilobase pair. So you can add only 2.5 kilobase pairs. But in case of if you remove some other portions, the size will be reduced from 48.5. So you can add more and more amount of DNA. So the lambda vectors are divided into two different types. One is insertional vectors and other is replacement vectors. In insertional vectors, the actual vectors is shown here. The A is an insertional vector, and this is the maximum size, that is 51 kilobase pairs. So the size of the real vector is just above 37 kilobase pairs. So what we do, we will add DNA, foreign DNA into this cell. RS is a restriction site. So this blue thing is the foreign DNA. We can add the foreign DNA into the vector, and it can go up to 51 kilobase pair. So the minimal size before addition the DNA, before inserting the DNA, the size should be 37 kilobase pairs. After addition, the size should be 51 kilobase pairs. So what we can add is 14 kilobase pairs of DNA can be added into the insertional vectors. Here we have another type of vector, is called a replacement vectors. The replacement vector, there is a stuffer fragment. It's like a fragment which is used to stuff, because only if the size is only below 37 kilobase pairs, it cannot be packed into the lambda head. So the size should be 37. In order to maintain that, we are putting some junk DNA or the stuffer fragment, stuffer DNA into the lambda genome, and during the time of recombination, we will be removing this stuffer fragment and replacing it with the DNA of interest. So that's what the replacement vector do. So the carrying capacity of replacement vectors used to be higher than the insertional vectors. In the insertional vector, we are just inserting, so minimum size is 37. So what we can do is up to 551. But in the case of replacement vector, its size is 37, but we can remove another portion from the genome and add our DNA of interest. Here you can see the insert size is this much in the insertional vectors, but insertion size is more in the replacement vectors. Here are some examples of the insertional vectors. Wild type phage can be used. Another one is lambda GT10, then lambda SAP2. These are insertional vectors. And we have replacement vectors. Here you can see the stuffer fragment is getting replaced by the DNA insert. Lambda B1 is an example, and more commonly lambda EMBL4 is an example of the replacement vectors. So here it is. So single recognition site for one or more resistant enzymes is the feature of the insertional vectors. So as we see, lambda GT10 is an example, and Karen 16A an example. So this insertional vector, its size is about 42, I think 42, about 43. Okay, and it has a left arm and a right arm, and in the middle they have a C1 gene. So in the C1 gene we have an eco-R1 site. So as its size is about 42, we can add about 9 kilobase pairs of DNA into the lambda GT10. And this C1 gene, which is actually the gene responsible for the lysogenic cycle. So if you add some foreign DNA into this gene activity will be reduced, and it will go for a Lytic cycle. Another example is Karen 16A. Instead of the C1, this Karen 61 is having a LAC-C gene, just like the PUC18. So to this LAC-C, we are adding we are inserting a DNA of interest. For replacement vector, they will be having a stuffer fragment, and we will cut both of them in this in both sides. We will cut and remove the stuffer fragments, add our DNA of interest. Classical example for that one is EMBL4. The carrying capacity is more for EMBL because we are removing the stuffer fragments. And another one is Karen 14. So the Karen 14 is having a polystuffer. The difference is like in this stuffer we will be cutting here and here, but in polystuffer there is multiple recession nucleus recognition site in the stuffer fragment, so it will be sliced into small pieces. And the advantage is that if you are cutting only here and here, there is chance that it will re-anneal to the original DNA when we are doing the ligation process. But if we are making it small pieces, the chances are slim. One advantage of lambda phage is like identification of the affected cells is easier than the plasmid vectors. One thing is that the size limitation. So if the DNA size is between 37 to 51, then only the packaging will happen. So if in some cases, if the DNA is not taking up the DNA of interest, its size may go down to the lower value than the 37 kilobase space. Then the packaging do not happen. If packaging do not happen, it do not produce an infectious virus, so there won't be any plaques. So that's one factor. Then, as I said before, inactivation of the CA gene. In some vectors, we are putting CAG gene in the middle. So if the CAG gene is inactivated, then only the lytic pathway begin. So if the CI or C1 gene is not inactivated, it will still in the lysogenic phase or something like that. And another factor is blue-white screening is possible. Just like the PUC18, we can do the blue-white screening for the lambda phages. One more feature I would like to talk about the lambda phage is the in vitro packaging. So lambda phage, still we are the science community is confused whether the virus is live or not. But one we can do is like in vitro packaging for the lambda phages. The process which is happening inside the cell, the packaging, the lambda DNA is attaching to the structural proteins and getting a functional virus, functional variant. We can do the same thing in the test tubes. If you have the DNA with the host site along with the head and tail proteins and other accessory proteins, we can actually make the infectious virus. So what we are making, what we are putting in the test tube will be the DNA and the proteins. But what we are getting after the packaging is the infectious virus. Just making artificial life, we can actually make the infectious viruses in a test tube. Maybe people will be trying to make the bioweapons like that. Okay, that's also possible. It's a very efficient transformation. The thing is that if you are packing our DNA into a viral genome, virus has an efficient method to transferring the DNA into the cell. So if our genes are in the inside the phage head, it will efficiently transfer the DNA into the cell. So efficiency of transformation will be high. The requirements is the DNA with a size between 37 to 51. That's the capacity of the lambda head, and the DNA should have a cos site. Then only it will be cut and inserted into the head. And finally, we should have the phage structural proteins. So we are making the phage structural proteins in a two-step procedure. What we do, we have two different lysogens. One is called a mutant E and other is mutant D. So mutant E is produced by a lambda phage called BHP2688, and the mutant D is actually produced by BHP2960. So the peculiarity of mutant E is like it do not produce E protein. So if there is no E protein, there won't be any head. But it is able to produce tail and D protein. Okay, so tail and D protein produced by this cell. So there is no functional virion, there is no head, so it do not produce a functional lambda phage. But the other lysogen is the mutant D. It do not produce D, so packaging do not happens, but it's actually producing head and tail plus the E protein. So E protein is produced by the D mutant and the D protein is produced by the E mutant. So you'll be collecting these components from each of them and mixing it along with the coarse DNA, the large DNA. So what's happening here is like the DNA is getting packaged to the head and the functional virion will be produced, because this process do not need the presence of a living cell. You can do it in the test tubes. So the advantage is that the efficiency of transformation is very high. If our DNA is packaged into a viral cell, virus know how to put the DNA into the cell, and it is also able to make the plaques. So it's a wonderful way of making viruses as a tool for transformation. As you know, some of these corona vaccines are delivered by adenoviruses. So this is another figure for in vitro packaging. The same thing is here. This head protein, then there has some defect, then the DNA is added. After the head is completed, the whole virus, infectious virus, come. So that's all for the lambda vectors. In the next session, we'll be talking about more type of viral vectors. Thank you so much for listening.